In this video, we're going to talk about the biology of Trypanosoma Brucei. In the previous audio, we talked about the family Trypanosomiasis, and we're able to mention that these Trypanosomiasis are also known as they are under the group of zoomastigophore, and they are flagellates, which means their organelle of locomotion is flagella. And we mentioned that they have their flagella that arises from the kinetoplast, and they reproduce by asexual means, and the reproduction is by what? Is by longitudinal binary fission, right? And we were able to mention that they have various morphological form, and part of the morphological form is a mastigote, which is characterized by having a central nucleus and lacking a protruding flagella. They have the promastigotes that have a cylindrical body, central nucleus, and anteriorly placed kinetosome, where the fluid flagella arises from. We have the choanomastigote that has a central nucleus, a kinetosome placed in a cylindrical-like body, where the flagella arises from. And we have the epimastigote. The epimastigote that have an undulating membrane, The epimastigote that is characterized by having a short undulating membrane, anteriorly, or let me say almost anteriorly placed kinetosome, and they have a central nucleus as well. And we have opistomastigotes that has posterior kinetosome, central nucleus, and they have a free flagellum, but they lack undulating membrane. We talk about the trypomastigote as well. Trypomastigote, which is the infective form of Trypanosomiasis, which has a posterior kinetosome, a retracting flagella, and from this kinetosome or kinetoplast, they have what? They have a free flagellum. They have an undulating membrane that is formed by what? By the retracting flagella. And we mentioned Trypanosoma species, and from there we said we have two types of trypanosome. We have the anterior station trypanosome and the posterior station trypanosome, right? And I said anterior station trypanosome is called what? Salivarian. The posterior station trypanosome is called what? The stercorarian. And this trypanosome is capable of what? Of infecting both man and what? And other mammals like cattle, and they cause a sickness known as African sleeping sickness. Now, today we'll be talking about Trypanosoma Brucei. Now, what are Trypanosoma Brucei? Trypanosoma Brucei is a species of parasitic kinetoplastid belonging to the genus Trypanosoma that is present in sub-Saharan Africa. This means you can mostly find Trypanosome species in sub-Saharan Africa, or let me say Africa generally, you understand, because they cause a disease known as known as African sleeping sickness. And they are named as kinetoplastid because they have what? They have kinetoplast and kinetosome. Unlike other protozoan, parasites that normally infect the blood and tissue shell, it is exclusively extracellular and inhabits the blood, plasma, and the body fluids. It causes a deadly vector-borne disease. Like we know, what is the vector-borne disease? Known as African sleeping sickness. You understand? Now, this trypanosome, like I said, it causes a disease known as what? African sleeping sickness. It now has, we said Trypanosoma Brucei, right? Trypanosoma Brucei. And remember, in the system of naming, we have the generic naming and the specific name. We have the generic name and the specific name. The generic name that always starts with capital letter, and the specific name that always starts with small letter, right? And if you notice very well, in this, we have the generic name already and we have the specific name already, which is Trypanosoma, that is the generic name, Brucei, which is the specific name. We now still have subspecies under this group of animals. So we have three complexes. We call them species complex. Now, these species complex are three. We have Trypanosoma Brucei, Trypanosoma Brucei, Trypanosoma Brucei gambiense, and Trypanosoma Brucei rhodesiense. These are the three complexes that we have. So I repeat: Trypanosoma Brucei, Trypanosoma Brucei gambiense, and Trypanosoma Brucei rhodesiense. Now, the parasite is a parasite of non-human mammals that causes nagana. They causes nagana in non-human mammals like cattle, you understand, and they causes sleeping sickness in mammals, you understand. Now, let us now talk about each complexes. Talking about each complex, let's talk about Trypanosoma Brucei gambiense first. Trypanosoma brucei gabesis causes a slow onset chronic trypanosomiasis in humans, and it is most common in Central and Western Africa, where humans are thought to be primary reservoir. So, in this, what you have to note is that humans are primary reservoir of what? Of Trypanosoma brucei gambiesi. Are you getting me now? There are several reports showing that animals, including cattle, are also infected with this group of animals. But 98% of human African trypanosomiasis is caused by what? By Trypanosoma brucei gambiesi. So we can basically say it is mainly found in what? In man. You understand? It is mainly found in man, and it is transmitted by a vector known as tsetse-fly. Tsetse-fly has a botanical name Glossina sp. So we can name it. Let me write it here. Glossina sp. You understand? These are Glossina sp. So we now have different type of Glossina species. We have Glossina palpalis. We have Glossina taxinoides. These are the two Glossina that I know that transmit what? That transmit Trypanosoma brucei gambiesi. Now moving from there to Trypanosoma brucei rhodesiense, they cause onset acute trypanosomiasis in humans. A highly zoonotic parasite. It is prevalent in southern and eastern country where animal games or game animals and livestock are thought to be primary reservoir. You understand? So, Trypanosoma brucei rhodesiense is found in what? Is found in both humans and animals. You understand? It is found in both of them, and it can lead to acute disease in man and can also lead to death. And it is transmitted by a Glossina species known as Glossina pallipedis and Glossina siwatoni. You understand? And I think Glossina mosatan. These are three Glossina that can transmit Trypanosoma brucei rhodesiense. Now move from there to the next one, which is Trypanosoma brucei. Trypanosoma brucei causes animal trypanosomiasis along with other several species of Trypanosoma. Trypanosoma brucei is not infective to humans, so you have to know that Trypanosoma brucei is not infective to humans, but it is infective to what? It is infective to animals. You understand? It is infective to other mammalian animals. So this species is really infective to humans, like I mentioned, and it is transmitted by Glossina mosatan and Glossina pallipedis. These are the two Glossina that transmit what? That transmits Trypanosoma brucei. Right now, moving from there to how can I distinguish Trypanosoma brucei from Trypanosoma brucei gambiesi from Trypanosoma brucei rhodesiense? You understand? The subspecies cannot be distinguished by their structure because all of them have the same structure. But how can I distinguish them? I can distinguish them by geographical location. You understand? Is the main distinction. And molecular markers are also developed some individual identification, like cell resistance associated genes. It is used to differentiate Trypanosoma brucei rhodesiense from other species of Trypanosoma brucei. You understand? Because there is a specific distinction in the specific distinction in the strain. Remember, I told you that molecular marker, so it is used to mark some molecules like DNA molecules in them. You understand? So we can use this to what? We can use tools to distinguish them. Are you getting me now? So that is how to distinguish each species. Are you getting me now? Now, moving from there to polymorphism, we can say the group of Trypanosoma brucei are morphologically distinct. I repeat, the group of Trypanosoma brucei are what? They are morphologically distinct because they exhibit what we know as what? Polymorphism or polymorphic form. The polymorphic form of Trypanosoma brucei is found in what? In mammal host, and only one of them is found in the vector host. You understand? Only one of them. So we have three. We have three morphological form. The first one is slender form. That's the first one. The second one is intermediate form, and the third one is what? Slumpy form. Let's just talk about the slender form first. The slender form is thin. You understand? It is thin, and it measures about 20 to 40 micrometer in main length and 29 micrometer in average length. Are you getting me now? They have a free flagellum running posteriorly. They have a what? They have a free flagellum running posteriorly, and it has a located kinetosome. They are found in the early stage of infection. So the slender form are found in what? In the early stage of infection. Now, how does slender form looks like? They look like normal regular trypanosome. Like I said they are thin and what? They are thin and slender. So they look like this. This is how they look like. They are thin and slender. They have an anteriorly located kinetosome. they have an undulating membrane, and they have a retracting flagella. So that's how they look like. So let me just clean this line. So these are how they look like. I get me now. So they have a central nucleus. So this is how the slender form looks like. That's the first one. Now the intermediate form. The intermediate form is between the slender form and the stumpy form. You understand? It is moderately long. It is what? Moderately long, and um, it usually measures about 20 micrometer in length and has a moderately long flagella. Are you getting me now? Moderately what? Long flagella. It uses a short period before it develops into stumpy form. Are you getting me now? So let me use this structure to denote him. You understand? So it is intermediate. It is not very long. Are you getting me now? It has an anterior flagella. It has an undulating membrane and a very short flagellum. Are you getting me now? And it has a central nucleus. So this is how an intermediate form looks like. What about the stumpy form? The stumpy form is usually short but big. They are what? They are short but big, and it is measured about 15 micrometer in length and 18 micrometer wide. They devoid flagella, but if the flagella is even present, it's going to be reduced. And they are only featured in what? In the parasite vector. So you found the stumpy form only in the what? Only in the parasite vector. So it can come in different morphology. It is what? It is short but what? But big. Are you getting me now? It has a kinetosome. It always diploid flagella, but if it has flagella, it's always what? It is always reduced. And it also has what? It also has a central nucleus. So these are how they look like. Are you getting me now? Now let us now talk about their life cycle. The life cycle of Trypanosoma Brucei, Trypanosoma Brucei, because all of them have similar life cycles. So let me just talk about Trypanosoma Brucei whole. Just know that if I'm talking about Trypanosoma Brucei, I'm talking about the three trypanosoma complexes, which is Trypanosoma Brucei, Trypanosoma Brucei gambiense, and Trypanosoma Brucei rhodesiense. You understand? Trypanosoma Brucei is heterogeneous. When you say something is heterogeneous, it means they have indirect life cycle. And when you say something has indirect life cycle, it means the life cycle involves two hosts. And which are these two hosts is the parasite vector and the mammalian host. So the parasite vector is known as the Glossina sp or Glossina species. Are you getting me now? And the mammalian host can be cattle or man. Are you getting me? So they cause a disease known as African sleeping sickness. The life cycle begins with the vector parasites. With the vector parasite taking a blood meal. You understand? They take a what? They take a blood meal from the what? From the mammalian host. And this blood meal that is taken is the stumpy form of trypanosome. Remember I told you there are three forms: slender, intermediate, and stumpy form. So they take the what? They take the stumpy form, and this stumpy form now enters the midgut. Once it enters the midgut, within 10 days, it finds its way to the posterior of the midgut, like the starting of the midgut. Are you getting me now? Where it divides into a slender procyclic form. Are you getting me now? It develops into what? Into a slender procyclic form. This then breaks to the peritropic membrane, separating the gut from the body wall. You understand? There's something known as peritropic membrane. It separates the what from the gut from the what from the body wall. Are you getting me now? Then from here, it uses a channel to migrate to uh, it uses a channel to migrate to the foregut. I'm sorry, not foregut. It is foregut. Foregut. Now, in this foregut, it developed into a mesocyclic form. Remember, I told you there are procyclic forms first, Abi, from procyclic to what? To mesocyclic. So the mesocyclic then divides. It divides and finds its way to the salivary gland. So it finds its way to what? To the salivary gland, where it transforms from the mesocyclic form to what? To epimastigote. Remember what I told you about epimastigote. How does epimastigote look like? Epimastigotes also look like the trypomastigotes, but the only thing is that the kinetosome is almost anteriorly placed. Are you getting me now? So the epimastigote then develop into a metacyclic trypomastigote. I repeat, metacyclic what? Trypomastigote. Remember what I just said now? I said epimastigote looks like trypomastigote. So from epimastigote, it now develop into what? A metacyclic trypomastigote, which then divides to form a what? A stumpy form trypomastigote, which means inside the vector host, it's now developing into what? Into a stumpy form trypomastigote. This stumpy form is now injected into the what? Into the blood mammalian host. Into the mammalian host blood. Are you getting me now? During blood meal. So another host, they inject another stumpy form into the host. Now, it should be infected. It should be noted that this disease is transmitted by what? By biting. You understand? By injection or arthropod what? Arthropod biting through their what? Through their buccal cavity. So this stumpy form that is injected into the bloodstream undergo polymorphic development. It undergo what? Polymorphic development. And what is polymorphic development? From slender form to intermediate form. From intermediate form, then it develops into what? Into a stumpy form. Are you getting me now? Then from that stumpy form, the what? The another vector parasite comes, takes the stumpy form, and the life cycle continues like that. So let me show you a visual this thing of the life cycle. So this is how the life cycle goes. This is how the life cycle goes. If you notice very well from this life cycle, the what? The fly takes a what? A blood meal, right? Takes a blood meal from the what? From the host. Are you getting me now? So it injects a what? It injects a stumpy form in the what? In the bloodstream of the host. Are you getting me now? So it develops. It undergo binary fission in the blood. So it develops, and remember I told you that trypomastigote. So inside that trypomastigote, it's developed into another stumpy form, which continues like that, you understand, in the bloodstream of the what? In the bloodstream of the mammals, you understand. So this is what? This is the stage. This is the life cycle. Are you getting me now? So inside the mammalian host, inside the parasites, this is how things looks like inside the gut of the parasite. I'm just going to visually illustrate it. So we have something like this. Then we have another thing like this. Then from there we now have the foregut. We have the foregut. We have the midgut, and we have the hindgut. You understand? So this is the foregut, this is the midgut, and this is the what? Hindgut. So let me just illustrate that. Foregut, midgut, and what? Hindgut. So remember, it is inside this midgut that we have the what? That we have the stumpy form. So the stumpy form comes to this midgut. Then it now find a way into the what? Into the peritropic membrane. So this is where the peritropic membrane is. So it find a way into the peritropic membrane. So this is with the peritropic membrane, so that it can be transmitted into the what? Into the foregut. I get me now. And once they enter the foregut, there is a salivary gland around this place. This is the salivary gland. They find their way to the salivary gland, and once they get into the salivary gland, the stumpy form gets into the salivary gland, then it is going to be injected into what? Into another human host. So this is an human host. You understand? Into another human host, and the life cycle continues. So this is the what? This is the form. Now, let's now talk about their pathogenicity. Their what? Pathogenicity. Now, the infection occurs in two stages. I repeat, infection occurs in two stages, which may be sometimes preceded by development of Trypanosoma Brucei on the site of inoculation within days of being bitten by an infected what? By an infected parasite. Now, it's the first stage of this disease is known as hemolymphatic. When you hear the word hemo, it means blood. So hemolymphatic, which involves non-specific signs and symptoms such as intermittent fever. When you hear the word intermittent, it means irregular fever, pruritious, lymphadenopathy, and the posterior triangle cervical lymphadenopathy or winter-bomb sign is commonly seen in Trypanosoma Brucei gambiaceae infection. But lymphadenopathy can be seen in axilla, inguinal, and epitrochlea regions. Enlarged lymph node is also a symptoms, and it tends to be submandibular, axillary, and inguinal in Trypanosoma Brucei rhodesiense. Now, the second stage of this disease is known as meningoencephalitic. That is invasion of the central nervous system. They are capable of invading the what? The central nervous system, causing what? Causing drowsiness, mumbling of speech, tremor of the tongue, and patients may become comatose, and there is sometimes sign of heart failure. You understand? Sometimes sign of heart failure, and if it is not treated, it can cause what? It can cause death. It can lead to death. And this is what is the pathogenicity of what, or pathology of Trypanosoma brucei complex, Trypanosoma brucei complexes. You understand? So we have, I already talked about this. We have the short stumpy form and the long slender form. Are you getting me now? And that is all about Trypanosoma brucei. Thank you for listening and subscribe for more.